I think we're going to come up with different drugs and different cocktails of drugs that will show signs of not reducing senescence, but actually using transcriptome, the lipidome, the metabolome, all the different omics, make a cell look younger. So could you engineer our genome to carry variants that have been shown to lead to extend the lifespan in humans?
Dr. Robbins, welcome to the show.
Great. Pleased to be here, Siim.
Yeah, I'm happy to have you on the podcast. Now, many people might not be aware of you, but you're pretty known in the longevity field. So maybe you can give a short and brief introduction about your background and what is your main area of research.
Yeah, great. Yes. So I actually started in the gene therapy field. So my first job as assistant professor was University of Pittsburgh, focused on gene therapy for a variety of diseases. I ran a vector core facility in addition to running the Molecular Medicine Institute. But as we were developing therapies for a number of different diseases, I started to realize that the root cause of many of these diseases were similar, and they seemed to be things that change with aging. So I very quickly bought into this geroscience concept that the greatest risk factor for most diseases is the aging process. So when I moved to the Scripps Research Institute in Jupiter, Florida, we started to focus on ways to target aging therapeutically, choosing cellular senescence as the hallmark of aging, which we thought was the most druggable at the time. And this was more than a decade ago. 6.5 years ago, I moved to the University of Minnesota, where I'm the co-director of the Masonic Institute on the Biology of Aging Metabolism. And we're very focused on cellular senescence, but a few other hallmarks of aging as targets to try to extend health span, not necessarily lifespan.
Gotcha. Yeah. Senescence is a synonymous to aging. And you mentioned one of the hallmarks of aging is cell senescence. So what is cell senescence in the human body and other organisms? Like, what kind of a phenomenon is it, and how does it relate to aging?
Right. Great question. So cellular senescence, or it's senescent cells, it's a cell fate. So this what happens to almost any cell in your body if it acquires too much damage. So this could be genotoxic stress, it could be oxidative damage, it could be mechanical stress. And if these cells then are stimulated in the way that they don't like, what they do is they exit the cell cycle and they undergo what's known as cellular senescence. And what your body wants to do is get rid of these cells. So these senescent cells, in many cases, not all, start to release inflammatory factors. And this stimulates your immune system to come clear them. So seeing somebody of your young age, your immune system is probably clearing thousands or tens of thousands of senescent cells a day. Does it very effectively. But as we age and our immune system starts to fail and you accumulate more cells, there tips a balance where our immune system doesn't clear them. And this leads to an increase in inflammation, but other adverse effects that are caused by this chronic accumulation of senescent cells. But cellular senescence is thought to be an anti-tumor mechanism. So one of the ways that the cells are induced to enter a senescent cell state is by activation of an oncogene, a mutation in the gene or protein that drives cells to replicate. So when cells start to replicate when they're not supposed to, this senescent cell fate kicks on. Cells exit the cell cycle, enter a state of cellular senescence, secrete these inflammatory cytokines, and the immune system clears them, and that reduces our risk of cancer. So senescence is a positive thing, but as we age and these cells accumulate, it can drive adverse pathologies and diseases related to aging.
So there is every cell has its, I guess, expiration date or a lifespan, and one of the outcomes of that journey is a cell senescence.
Well, I don't know if they all have necessarily a lifespan. If a cell, and this can be a fully differentiated non-dividing cell, such as a liver cell or a kidney cell or a brain cell, if they acquire so much damage that this cell fate then is stimulated and takes over. So some cells never senesce. Other cells, if they have damage, will senesce. The other thing is that a senescent cell can drive neighboring cells or cells at a distance to enter a similar state because of these inflammatory factors that they secrete. So one senescent cell can drive additional cells to enter the senescent cell fate, which then amplifies the adverse effects. Should also mention that senescence can play a positive role. There are examples where senescent cells come up or arise acutely in cases of wounding during embryogenesis. And so it's thought that some of these kind of transient senescent cells actually release good factors that can stimulate wound healing or facilitate embryogenesis or some other positive effects when we're young and healthy.
Mm. Yeah. So it's like the aging process, aging is characterized by many things, one of them being inflammation and mitochondrial dysfunction, like these other hallmarks of aging, and immune dysfunction as well as you get older. So your cells become more vulnerable to this, let's say, damage, like colloquial term, damage, and then the damage, the cell can decide, okay, we can either turn cancerous or we can turn this cell senescent. And the, anti-cancer, more rational approach would be to turn it senescent. So it's a better outcome than turning it cancerous. But both of them still lead to a decline in health over time, because as the cell senescence accumulates, then you're experiencing, I guess, faster aging, as I understand it correctly.
Right. And so as you probably have heard from others, these hallmarks of aging, which have been identified, are all linked. So senescent cells have dysfunctional mitochondria. There's a lot of evidence that dysfunction in the mitochondria plays a role in driving cellular senescence.
Hmm.
Another example is that stem cells in your body, as they age and acquire damage, may become senescent. So you lose stem cell function. And obviously, senescent cells release inflammatory factors, so they contribute. To what extent, we're not quite clear, but they contribute to inflammation. So
Hmm.
senescent cells have many of the hallmarks of aging in them. They have altered proteostasis, altered autophagy, different changes in epigenetic marks. So a senescent cell has many of the same features that affect the whole body and our other hallmarks of aging, because they're all linked.
Gotcha. So it's not like some chicken or egg
Right.
problem. Or it is a chicken and egg problem.
Yeah. And if, so when senescent cells have dysfunctional mitochondria, that leads to increases in reactive oxygen species, which causes more DNA damage. This causes oxidative DNA damage. And then that leads to the stimulation of more of a senescence state. So there's this autocrine effect. There's also a paracrine effect. Like I said, it affects neighboring cells and drives them into a state of cellular senescence.
Hmm. You mentioned that in early life, these senescent cells can be beneficial for wound healing.
Right.
Um, I guess the question is, like, in older people, if they have good wound healing, would it be somehow pathological, or would it somehow be detrimental because it would be like a sign of higher amount of senescent cells, or
Right. It's a great question. I think the field's still trying to determine what a good senescent cell looks like, how to characterize it versus a bad senescent cell.
Mhm.
Um, but I think the data is suggesting that in older people, that clearing senescent cells, and that may be predominantly the chronic ones, actually improves wound healing. When
Okay,
you're young and you get rid of these senescent cells, it seems to inhibit wound healing. But when you're older, getting rid of the bad cells seems to be beneficial. Um,
gotcha.
and clinical trials will be determining that. I think trials are currently ongoing, so we will see if wound healing, whether it's diabetic ulcers or if it's recovery from surgery, the wounding from surgery, or other types of events, if
Hmm.
clearing senescent cells in older people improves those processes. And
Hmm.
my guess is the answer is going to be yes.
Gotcha. What about lifespan in animals? Does clearing them out improve lifespan?
Yeah, a great question. So there's, I won't say conflicting data, but there's some experiments, and this has mostly been done in transgenic mice where they've been modified so that you can clear with treatment with a drug certain types of senescent cells. And in certain cases, clearing those cells using these genetic models does not lead to a dramatic extension of lifespan. There was a recent paper that said that clearing a subtype of these senescent cells, these are ones that have a marker called P21, extend the lifespan by about 10%. And there's a little bit of evidence with drugs, which we term senolytics, that will preferentially kill senescent cells, that there may be a slight increase in longevity. But what we're looking for, I'm not a big believer that we're going to extend lifespan by 50 years over the next generation. The goal is to try to keep people healthier for longer. So we're
Mm,
really focused on health span. And I think one of the side effects of extending health span is that there is an increased lifespan.
gotcha.
What
Yeah.
we don't want to do is increase lifespan so that the same decrease in vitality, accumulation of frailty still happens at the same rate when you're just a little bit older. What we want to do is compress the curve so this period of comorbidity is very short as
Mm,
we age. So we're healthier for longer and then fall apart quickly.
right.
And now you have that slow decline that we're seeing that leads to time in nursing homes.
Yeah, people are living longer on average today than in the past, and that can be a negative thing because if you have a lot of old people who require a lot of help and medical expenses, etc., then that can overwhelm the system.
Yes.
Yeah, the goal is to have people who do live slightly longer, but more importantly, they stay healthier for longer, and they have this longer health span that they don't accumulate a lot of these comorbidities late in life.
Yes, and there's a lot of discussion, and you may have had guests on your program that may argue that we're going to see this huge extension of lifespan, and
Mhm.
uh, I'm not necessarily disagreeing with that, but I think we're a long way off to start seeing those effects.
Right.
Um, Again, I see what we're seeing in model systems, mice, rats, maybe non-human primates, is we can kind of extend this period of healthy aging, whether it's going to be a few years or longer. That I think is the initial goal. And we'll
Yeah.
talk about longevity in the future.
Yeah. Yeah. And senescent cells can obviously have a important role in that. Do does I would imagine then a higher amounts of these senescent cells correlates to more of these comorbidities like heart disease, cancer, Alzheimer's, and other health ailments.
Yes. So, the question is whether all ailments are going to be driven by, or at least have a contribution of senescent cells. I think in many cases, it appears that with human disease, whether it's osteoarthritis or if it's Alzheimer's or liver diseases, that there seems to be an increase in cellular senescence. And always the question is that the consequence of the disease or the cause of the disease? And I think senescent cells contribute, but it's not the only cause and not the only target. So, I think we will see benefits in numerous human diseases if we can reduce the senescent cell burden. Um, but it's not going to be the cure for everything. There's going to have to be other hallmarks of aging targeted, other modifications made to treatment protocols. But clearly, reducing senescence in older people that have an increase in senescence, and that's the other issue, is that we're not a, a mouse model that shares identical genomes. Everybody's different. And I think that if you took a room of 65-year-olds, there would be a third of those that would have high senescent cell burden and may not be aging as well. And then you're going to have a third that really have not much senescence. So we really have to identify ways of monitoring your senescent cell burden to determine if you're really one where a senolytic will work.
Right.
Uh, and so I think we're, biomarker development so we can identify markers of bad senescence, or, maybe there's a certain type of senescent cell that drives heart disease versus liver disease versus neurodegeneration. So I think
Mm.
trying to identify the senescent cells we need to target for different diseases is also going to be important.
I want to take a quick break to let you know about my favorite wellness devices, which are the bond charge red light therapy devices. You've probably seen many of your favorite longevity influencers using these red light therapy devices, but do they actually work? Yes, they do. Red light therapy has been shown to have many benefits on skin anti-aging, hormone optimization, pain management, and even exercise performance. I use my device every day for 15 minutes, especially during the winter months when there's not much sunlight. It increases my energy in the morning and makes my skin glow. Just check out the testimonials on the bond charge website for the before and after pictures of other people. Most red light therapy devices don't have the right wavelengths of light, which might mean you're not getting the claimed benefits. Bond charge uses the exact wavelengths of light used in research, and they also have near-infrared light that's beneficial to the joints. Head over to bondcharge.com and use the code SIIM, SIIM, for a 15% discount.
Are there any ways to eliminate these senescent cells, or is it actually something that you want to do
Right.
to remove them?
Yeah, it's a great question. I think the data in model systems says clearing senescent cells kind of across the board is beneficial,
Mm.
at least in older mice. Younger mice, that's a different question because they're, like I said, these cells may be beneficial. Um, so it appears that if you take older mice and you treat them with drugs, and I'll talk about these drugs in a second, and these are drugs that are termed senolytics, that will kill at least a subset of senescent cells, that there are benefits across the board. And this seems to improve immune function, seems to improve cognitive function, seems to improve liver function, seems to reduce inflammation, improve mitochondrial function, improve stem cell function. Um, so the drugs, the class of drugs we're working on are termed senolytics. And we were involved in the identification of the first senolytics with Jim Kirkland and Tamar Ciccone at the Mayo Clinic. And so we identified the first generation of drugs, which in most cases turn out to be anti-cancer drugs. And it makes sense because, as I alluded to, senescent cell fate can be driven by oncogene activation. So you have cells that are starting to become cancerous, senescent cell fate's turned on, and what appears that there actually are these networks or pathways that block apoptosis or cell death. The tumor cells turn on. That's one of the ways they become kind of immortal and able to proliferate in your body. And so drugs that target these pathways that keep the cells from dying, tumor cells, also appear to kill many types of senescent cells. So many of the drugs which have been approved for cancer seem to have effects at least on certain classes or types of senescent cells. Um, we've also identified natural products that have this ability to kill senescent cells, but many of these also have had the ability to kill tumor cells. So in some ways, we're kind of killing that early stage precancerous cell that may lead to a tumor cell using drugs which have this senolytic activity. So in theory, if we use our senolytics, it's going to clear these senescent cells that have oncogene activation that if they break through the senescent cell fate, they eventually lead to cancer. So in
Mm.
theory, senolytics will reduce your risk of most types of cancers as a way of extending health span and potentially lifespan.
Gotcha. Yeah, that would be like the best case scenario that
Yeah,
you'll eliminate both the senescent cell and the cancer cell.
yeah,
Which of these senolytics are the best researched?
right. Right. So the first generation, and this was the paper we published with Jim Kirkland in 2016, was showing that an anti-cancer drug that targets what are known as tyrosine kinase receptors, a whole family of them, that this drug, dasatinib, in combination with a flavonoid that's present in many fruits and vegetables called quercetin, that combination was able to induce what we call senolysis, or death of senescent cells in culture. And there probably have been 24 or more clinical trials either started or completed at this point with D plus Q, as we call it, dasatinib plus quercetin. There's evidence. I mean, it's not overwhelming evidence, but in some of the clinical trials that have been finished, there's evidence that in the top 35% that have the most senescent cell burden, that they're responding or showing benefits of D plus Q.
Mm.
I don't think dasatinib and quercetin is really that much of an optimal senolytic combination. It was just the first one identified. Dasatinib is an FDA-approved anti-cancer drug. Quercetin you can buy from Amazon and as a supplement. So it was easy to move into the clinic. But they're now what we call second-generation senolytics coming, multiple targets identified in different types of senescent cells. So I think there's going to be much better and much safer senolytics that are at least in preclinical models, and we'll have to see how they function in the clinic. I should point out, I keep talking about different types of senescent cells, but it's really becoming clear that if you take a tissue such as the brain or the liver, so for example, the liver, you can see senescent hepatocytes, senescent Kupffer cells, senescent infiltrating immune cells, and each one of these senescent cells seems to have a different inflammatory profile. So the thought is, and we haven't proven it, that they may be having different types of adverse effects. And so it may be for certain types of liver diseases, you want to clear senescent hepatocytes. For another type of disease, you may want to clear the infiltrating senescent immune cells. So we're still trying to sort out what are the different types of senescent cells that we need to target, and then what's the best drug or drug combination to do that.
Mm. Yeah.
And I guess is the other question you asked, is it beneficial to clear senescent cells? Because you're in theory making a hole in the tissue where these senescent cells are. And I think the best example is, if there's senescent neurons that accumulate with Alzheimer's or Parkinson's, and these cells don't replenish because they're fully differentiated. So killing off a senescent neuron may not be a good thing.
Hmm.
But at least in animal models, clearing senescent cells from aged mice or mice with neurodegenerative diseases like Alzheimer's, it's been beneficial. There's better cognition, better behavior, better motor skills. So we've not seen any adverse effects for eliminating a few senescent cells in the brain or in other tissues, at least at this point. We'll
Yeah.
have to see as we continue in the clinic with these clinical studies.
Yeah. I guess, the most important thing is what's the final outcome. There might be some side effects with clearing out senescent cells, but if you live longer and you're healthier, then it's apparently not that bad. Or, there's the end outcomes are something that I think matter a lot, sometimes more.
Right. And, it may be that, as we'll find out, that there really are not that many senescent neurons. So it's only a few that are being cleared. It appears that it's the microglia, which are immune cell type in the brain. They're similar to macrophages, that they develop a senescent-like phenotype. So clearing those appears to have a benefit. Maybe astrocytes, maybe oligodendrocytes. So we're still identifying the right senescent cells that are arising, what they are, where we see them in the brain, and then what drugs are the most effective at killing them. But again, as I said, in animal models, we've seen no adverse effects of clearing senescent cells,
Gotcha.
at least in older animals.
Yeah. Would it be possible to somehow reverse the senescent cell back to a normal cell or more youthful cell, kind of some gene editing with Yamanaka factors or something to make it more youthful again?
Yeah, that's a great question. So, you could discuss whether taking a cell that's been damaged, that may have acquired mutations or other changes that might make it detrimental if it started to proliferate again. There's been discussions that, that's the last thing we want to do is rejuvenate a precancerous cell. But it does appear that with OSKM, which are the Yamanaka factors, that you can take in at least certain types of senescent cells and make them look younger. And I think it's going to be something we're going to have to determine clinically is if these more youthful cells are, if you have ones which are maybe damaged and have mutations that make them more youthful, if that's going to be a bad thing. Maybe as we rejuvenate cells in the body, it's going to improve your immune function. And so your immune function might still be able to recognize these precancerous cells and clear them. So that's the other kind of unknown is when we treat mice with drugs like senolytics or other targeting other hallmarks of aging, if you improve immune function, you're going to reduce your senescent cell burden. So many of therapies out there might have indirect effects on senescence by improving immune function, which then leads to better clearance of your senescent cell burden. So there's maybe multiple ways that a drug could function to extend health span and reduce your senescent cell burden.
Yeah, you mentioned the immune system, that it's central to clearing out the senescent cells during youth. And with age, you see a decline in immune system function, which is one of the reasons there's a higher burden of senescent cells. Are there any, I guess, therapeutic options here to support immunity in elderly people?
Right. So there are obviously a lot of supplements that are being sold because they improve immune function. I think with very little evidence that suggests that's the case. Um, I, I think there are. And so we have a large grant from the NIH to look at ways of reducing immune senescence because we do know that T cells, B cells, macrophages, NK cells show profiles that make them appear senescent. Are they truly senescent, or they're senescent-like? But they're clearly dysfunctional. So if you get rid of these dysfunctional immune cells, will that improve the function of the remaining cells? And so we are doing experiments to look at that phenomenon and see if we can identify compounds, which may not have to be senolytics, but other classes. Can we improve immune responses to pathogen exposure and to cancer? And so we and others are working on that, and I think that at least the animal data looks very promising.
Hmm.
Um, and there's, there are probably trials in humans too. I think the question always comes is, as I'm alluding to, if you have senescent cells that are releasing inflammatory factors, those inflammatory factors alter immune function because it changes their ability to react to a pathogen or to a tumor. So clearing senescent cells from the liver might improve immune function. Alternatively, if you can target the dysfunctional, potentially senescent immune cells, it will then clear the senescent cells in the liver and make the liver better. So we're still trying to sort out if, do we need to target immune cells or non-immune cells that are senescent? And I think the answer is going to be both. I think, as always, it's going to be a contribution of both, just the overall senescent cell burden you want to reduce, and you want to try to reduce senescence in different classes of immune cells.
What about rapamycin? That's one of those pharmaceuticals that has been seen to improve immunity in elderly people.
Right. So it gets back to, what cells are being targeted by rapamycin. But clearly, that was the first drug really demonstrated to extend lifespan in mice. It's in a number of clinical trials in humans. I think dosing and when do you start treatment is still a question. But yes, rapamycin has been shown by a variety of groups and ourselves to actually reduce senescence. It doesn't kill senescent cells, but it suppresses the inflammatory factors that senescent cells release. So drugs like rapamycin that suppress this inflammatory component, we call senomorphics. So it doesn't kill the senescent cell, it suppresses that. What we don't know is what the key target is. Well, not target, but which hallmark of aging is the target for rapamycin. Clearly the target's mTOR, that regulates nutrient signaling, regulates gene expression, regulates this inflammatory component in senescent cells. But is that how rapamycin is working? Is by reducing the inflammatory component of senescent cells? Is it improving responses to nutrients? Is it affecting metabolism? Is it affecting stem cell function? I think we're still sorting out, as in many cases with drugs, what's the key pathway. But it does reduce senescence, and I think that contributes. And through that reduction in senescence, we think improves immune function also. And that was shown by Joan Manick, where she treated a number of the older patients and showed that they responded to either a tetanus vaccine or the flu vaccine much more efficiently if they got intermittent rapamycin treatment.
Yeah. The previous belief was that rapamycin is an immunosuppressant drug that is used for organ transplant patients. But that turns out, if you dose it intermittently, then it actually increases the immune system function.
Yeah, intermittent treatment is in the lower doses, so the dosing is also important. Rapamycin has some side effects at high doses or continued treatment. There are a number of what are called rapalogs being developed, which may target more the right mTOR complex and not other mTOR complexes that might be beneficial. So we'll have to see. I think a number of these trials have started for different age-related diseases, and we'll see if the rapalogs are as or more effective than rapamycin. But clearly rapamycin has a beneficial effect if dosed correctly in mice and appears in people with certain conditions.
What would be then more, let's say, practical things people can do to reduce their senescent cell burden? And obviously it's going to be different between younger individuals and older, so maybe we can do, let's say older than 50 and younger than 50.
Right. So, unfortunately, the answer is the same answer you would get when you ask, what can you do to live longer and healthier? And that's diet and exercise. It's been shown that exercise in mice, and I think in humans, can reduce senescent cell burden, at least with the markers we have to measure senescence in humans. So exercise has, whether it's through stimulating your immune function to clear the senescent cells, or if it's through a different mechanism, still not quite clear. Your diet can affect senescence. We had a review article in Cell Metabolism recently with Dudley Lamming talking about all the ways you can reduce senescence and a variety of dietary interventions, which will do that. I think caloric restriction has a benefit there too. But not everybody, as we know, at least in the U.S., exercises or eats well. So we still think there's a role for developing pharmaceuticals that can target senescent cells that can be taken intermittently and safely to reduce the burden. And in mouse studies, senescence, diet, or a senolytic seem to give similar results, with in many cases, though, the senolytic working slightly better. So diet, exercise, and hopefully eventually we'll have compounds that you can take safely. And
Gotcha.
I think the reason why they'll be safe is because you take them intermittently. You don't have to take them daily to eliminate your senescent cells.
Yeah. What kind of dietary components did you find were beneficial?
Well, there's numerous studies. It depends on the model system, but it's clear that reducing your caloric intake, compressed eating, has a benefit, and then amino acid or diets that are reduced in certain amino acids that have altered effects on senescence. But it's still not clear what your right mixture of carbohydrates and proteins and then amino acid composition in humans is going to have the biggest benefit, because we see differences between mouse strains. So even within mice, different strains respond differently.
Gotcha.
So I think what we're going to need is a way of, like I said, really determining your senescent cell burden and determining if you need a certain type of diet or treatment as compared to somebody else. And so eventually this will become a personalized treatment regimen. You can either, send your blood in and get your epigenetic clock, or they can measure 5 senescent or proteins that senescent cells secrete, determine your senescent cell burden, and then make suggestions on either diet, exercise, and/or drugs you could take that target your senescent cells.
You mentioned the amino acids. Is it that certain amino acids increase cell senescence, or protein restriction or amino acid restriction reduces them, or which way is it?
I think it depends on the strain, probably depends on the cell type and the tissue.
Okay.
But clearly, I think if you look at mice, drugs that have the biggest effect on lifespan, there's rapamycin, but then there are metformin, SGLT2 inhibitors, and these are ones that seem to be used or are used to treat diabetes because they normalize metabolism. So at least in mice, normalizing your metabolism is beneficial. And if you normalize your metabolism, we know metformin treatment, SGLT2 treatment, or inhibitor of SGLT2, that they reduce your senescent cell burden. So improving metabolism reduces senescence, or maybe these drugs reduce senescence, which affects metabolism. But clearly drugs that humans are taking for type 2 diabetes, metformin, SGLT2 inhibitors, are having a benefit on health span across the board, as well as able to reduce markers of senescence.
Interesting. Yeah.
And I guess the other drug I should toss in there, and maybe we can discuss this later on, is GLP-1 or GLP-1 receptor agonists. That there was a long discussion at the recent meeting on aging research and drug development in Copenhagen. Is GLP-1 or are GLP-1 like drugs our first anti-aging drugs? Because there's data coming out that GLP-1, Ozempic and related drugs can have effects on Parkinson's, Alzheimer's, heart disease, osteoarthritis, liver disease. And it's still not clear the mechanism. I'm still surprises me, but every time I pick up a journal, there's an article about Ozempic or related compound having a positive effect on the age-related disease.
Mm.
So they're saying, will GLP-1 receptor agonists be the first true anti-aging drug? And the panel of people from pharma, they all said yes.
Mm. Right.
Surprising. So, GLP-1 also normalizes your metabolism. Again, we don't understand all the mechanisms. There may be adverse effects on muscle function, which they're trying to develop combinations of drugs that don't reduce muscle mass and function when you take GLP-1 agonists. But I think they're going to eventually overcome that side effect, and these will be drugs that
Right.
people will take after a certain age to extend their health span.
How big of that effect comes from just the calorie restriction or eating less?
It's a question I always ask, and I think the answer is not quite clear. They think there are actually some disease modification that, the best example would be osteoarthritis. Is that simply losing weight allows your knee joints to feel better. But they say there's actually changes in the joint architecture. It almost looks like a disease-modifying drug that can improve the joint structure. So, and that's thought not to be just through weight loss, but is it caloric restriction, mimicking caloric restriction mimetics? Yes, that probably is part of it, but they think there's more. And what's interesting is that many cells in the brain, chondrocytes in the joint, actually express the receptor for GLP-1.
Yeah.
Which again, I'm very surprised at why these cells would express a receptor for something that regulates appetite.
Yeah.
Still a lot to learn about GLP-1.
Yeah, people have the impression that it just makes you eat less, but there's a lot of other systemic effects because of the hormonal effects you get from that increase in GLP-1. So yeah, it's probably a lot of the effects come from eating less, but there's additional benefits as well, probably.
Yes, I think that's definitely the case.
Yeah. What about the these over-the-counter supplements you mentioned, fisetin, quercetin, apigenin, etc.? Is it something that people should consider already, or like what do you think is the evidence right now?
Right. It's a great question. I'm asked this one a lot because, again, in collaboration with Jim Kirkland, we were the first group to publish that fisetin had senolytic activity. And this has led to multiple clinical trials with fisetin. Fisetin is not the best senolytic, and it takes high doses to see a benefit in mice. We have made analogs of fisetin, which we think are much more effective, that we hope to be taken to the clinic soon. We've identified some of the targets of this class of flavonoids that probably bind or interact with 50, 100, or hundreds of proteins. So we don't really know what the key target is of fisetin. I think the problem with taking fisetin, and I don't think there's a downside. In fact, many people in the aging field are taking fisetin. I take it intermittently myself, but we really don't know the dose. So some people take a certain dose daily. Some people take a higher dose once a week. During COVID, when we showed that fisetin actually protects old mice from a virus similar to SARS-CoV-2, we started to take fisetin to protect ourselves from COVID if we were so infected. But again, we don't know what the right dose is. So we were taking for 2 days, we took high doses, went 14 days and another 2 days with a high dose. No evidence that that's the right dosing regimen. So I don't think there's an adverse effect of fisetin. At least we've not seen that, but I don't necessarily recommend people taking these huge doses every week or two. People that take it once a day have say they feel better. Quercetin's probably like fisetin, maybe not quite as effective, but there are a number of flavonoids. And another one that we're working on is not a flavonoid, it's a drug called fucoidan, which is a complex oligosaccharide that comes from brown seaweed that's taken as a health supplement in Asia. And there are dozens of different types of brown seaweed that you can isolate fucoidan from. But we showed fucoidan actually functions as we call a xenomorphic. It suppresses the inflammatory components of senescent cells. And we think it does this because it stimulates an enzyme called SIRT6, which is involved in DNA repair. And we have evidence in mice, at least, not in humans yet, but in mice, that fucoidan can improve DNA repair, which if you reduce genotoxic stress, there's less senescence, less inflammation.
Hmm.
And so they're now starting clinical trials with fucoidan to see if there's benefits in humans. Again, people take this, but there's been really no controlled studies to look at markers of aging, epigenetic clock, mitochondrial function, etc., and senescence.
Hmm. Yeah, SIRT6 is, I think, the only sirtuin that's been seen to extend lifespan in animal models. The other ones, not so much, but SIRT6, yes.
Yeah, so there are multiple companies now selling fucoidan as a SIRT6 agonist, and and I'm hopeful that the clinical trials that are being set up, that hopefully are going to be set up in a blinded manner, quality control, that will determine if fucoidan from certain sources indeed has a health benefit. Um, but people take fucoidan, they take quercetin, they take fisetin. Uh, we could talk about NAD precursors that people take. I think the data is still limited in humans about the benefit because we don't know the right dosing regimen
Mm.
or the subset of people that are going to respond best to these drugs. Not everybody's going to respond the same way across the board.
Gotcha. What about like gene therapies? They become, I guess, more known in the longevity space. And is there any even like a possibility to do some gene therapies to help with senescent cells? Or, I mean, I guess you can make gene therapies for immune system function, but what about senescent cells?
Right. So when you talk about gene therapy, aging, there are different types of gene therapies you could imagine. So, one of them being developed by Scott Lowe and Karina Moore at Cold Spring Harbor, they're making CAR T cells. So we can engineer T cells that will target a tumor cell. They're trying to engineer T cells to target a senescent cell. And they have shown if you clear senescent cells expressing a protein on their surface using these modified T cells, that the mice are healthier for longer. I think the issue is going to be is what is the right protein to target on a senescent cell? Is there really something specific for a senescent cell that's not expressed on normal cells? And so their question is going to be about safety moving forward. But I think there will be clinical trials using CAR T cells that target senescent cells in the future. Other people are trying to deliver growth factors using viral vectors. They can deliver to different tissues. For example, you can deliver clotho, which has been shown that mice without clotho live a very short lifespan. If you deliver it back with a gene therapy, you can extend lifespan of those mice. So that's another approach. I think the question there is if the protein you're expressing can have adverse effects if you make too much of it, there's going to be a safety consideration. So it may be certain gene products are going to be safe if you deliver and overexpress them in the body. Other ones may not be. If you make GLP-1 at an elevated level constitutively, is there going to be a downside in 5 years? So I think there's some safety considerations. The other approach, which has been discussed, and George Church, I think, has been a big proponent of this and has talked about it, is using CRISPR-Cas or other forms of gene editing to deliver variants that are found in centenarians into people that are not centenarians. And so I am part of a large effort with Albert Einstein and Columbia University to do sequencing of centenarian genomes to identify what we call not mutations, but they're variants. For example, we identified a variant in SIRT6 that make it more active, which led to us to do the fucoidan studies. So the thought might be that you could actually deliver or engineer certain cells and certain tissues to have this rare variant in SIRT6 or an IGF-1 receptor or in other, there's some in genes regulating cholesterol synthesis that may have effects on health span. So, so could you engineer our genome to carry variants that have been shown to lead to extend the lifespan in humans? I think we're a long way off from that, but it's feasible in theory. So we can deliver genes. Another example of a gene, keep talking about SIRT6, but it's kind of been something that my lab in collaboration with Vera Gorbanova and others that we've been focusing on. But we found variants in SIRT6 in centenarians companies have taken that centenarian SIRT6 and delivered it to liver with an AAV, an adeno-associated viral vector, and the mice are healthier. If you overexpress this variant of SIRT6, or you could engineer using CRISPR-Cas the gene edit in the liver to introduce this variant into the SIRT6 gene. So you could think about ways going forward, and I think we're a long way off because I think there's safety considerations. But it's something we could consider in the future that will allow those of us who are not going to be centenarians to live healthier for longer. The question is, how many of these rare variants do you need to take somebody who's not a centenarian? And I have no centenarians in my family, so I'm not expecting to live to 100. But can I engineer 5 genes, 10 genes, 50 genes? What, how many do you have to engineer for me now to be a centenarian? And in what tissues and what percent of the cells in that tissue? Lots of questions. It is something that companies are working on, and I think eventually we'll be there, whether it's 10 years or 50 years. I think eventually these approaches will be applied to the human condition.
Some of the ones we are aware of, things like ApoE4 and maybe IGF-1 pathways, maybe. And I would imagine there's something related to senescent cells as well, some genes to help them to clear them out more easily or something like that. There's immune system related genes that are associated with longevity. So there's a handful of them that we're aware of. I guess there's no one who has done all of them to actually see what's the effect.
Yeah, so it's always the question when you look at rare variants. You talked about IGF-1 pathway, SIRT6, and then you talk about bad genes like ApoE4. The beauty of the centenarian studies is there've been centenarians identified that have high risk variants in ApoE. Even people with BRCA1 mutations. That's something about these rare variants that they carry that are beneficial can block the adverse effects of a bad ApoE or BRCA1 or BRCA2. So, I think there's going to be a lot to learn from centenarians, and then obviously modulating IGF signaling, which is one of the first variants identified in centenarians. There's a transcription factor, FOXO3, that's been shown to be expressed differently in centenarians. So I think if we can identify the beneficial variants, they can actually suppress these bad variants. So you may not have to actually gene edit out your ApoE4. You may have to edit in a SIRT6 variant or an IGF-1 variant, or we some variants that affect the level of a transcription factor NF-kappa B, maybe downregulate NF-kappa B so it's not as inflammatory late in life. But these are things that will be coming as we sequence more centenarian genomes. We validate them in mice. We validate them in cell culture models. And then, eventually as the gene editing technology gets better and safer, I think these will be applied to trying to make us more of us centenarians.
Yeah. And then, I think this is going to be, one of the big leaps that will, lead to some more breakthroughs in longevity and life expectancies, because genetics is very powerful. I think a lot of people underestimate the power of genetics when it comes to how long you live, even just by the fact that if you have long-living parents or grandparents, then your likelihood of reaching that also like 100 or 110 or something like that is huge compared to the average person who doesn't have long-living parents. So yeah, longevity runs in families a lot. That it's very genetic, and if you understand those genes, then yeah, maybe we can use these gene editing to actually apply to more people.
Yeah, I think it's just going to be, the safety issue. Treating a rare disease that has a very poor prognosis with these new methods. I mean, that you can justify, but taking millions of people, across the country and starting to edit their genomes, wait 10 years and see what adverse effects there could be if it turns out there's off-target effects. So there's going to have to be a lot of studies done treating more severe diseases with these approaches to really support moving this into treating the masses. And there's also a cost factor too. I mean, the goal for all of us is to develop safe, effective, and affordable ways of extending health span. So again, we're not trying to do this for just people in Silicon Valley and Wall Street. We're trying to do this for, the average American and the average person around the globe. And so we have to do this. We have to do studies and characterize people with different ethnic background, which their genetics may be a little bit different. There may be different rare variants. Some may have a different ethnic background than another. But we have to come up with a way to make this kind of safe, effective, affordable for everyone.
Mm.
And that's the reason why something like a fisetin, if you can make a better fisetin or make, a natural compound or come up with a cocktail that's cheap and affordable. I mean, metformin is about the cheapest drug you can get. And if it really shows in the TAME trial to extend health span, that's a drug that many people could get access to.
Mm. What is your next biggest projects you're focusing on? Obviously we talked about the 10-year goal with a lot of these therapeutics. What is the biggest focus right now for you?
Yeah, it's hard to always pick one, but I guess I'll name three. We'll start with there. But so the first one is that within our institute, we've generated mouse models where we can induce senescence in the cell type or tissue specific way. So we're making a whole panel of mouse models where you can induce DNA damage and senescence in cells in the liver, the brain, and muscle, and the heart, and pancreas, the intestine, and then trying to look at what pathologies those subsets of senescent cells cause. Again, trying to identify what are the key senescent cell types we should be targeting. We're then trying to optimize our senolytics. We've developed in vitro cell culture screens. We have then mouse models that can validate the drugs that score positive as senolytics on certain types of senescent cells, and we're trying to optimize them. We're doing medicinal chemistry. So we really have a portfolio of new senolytics that we're trying to take forward, as well as identify what the right lead compound is. I mean, that's the problem is we have too many senolytics that look promising. We have to identify the one or two we want to take forward. And then we've gotten into trying to understand partial rejuvenation. Again, you asked about partial rejuvenation with the Yamanaka factors, which companies like Altos and others are really focused on. And whether, these OSCAM factors can rejuvenate senescent cells, and it appears in mouse models that they can. So we've been screening for senolytics that can take a senescent cell and not kill it, but make it not showing all these markers of senescence. And some of them actually make the cells look younger, healthier at the transcriptional level and the epigenetic level. So we're starting to think that some of the drugs we're identifying may have partial rejuvenation effects. Not only, and that's how they're affecting senescence is through partial rejuvenation and not by directly targeting senescence. So we are now trying to do more assays and more characterization, both in cell culture and in mice, about some of the drugs that we've identified that may have partial rejuvenation effects. I do think that what Altos and others are doing is very exciting. The question is always going to be safety. Are expressing Yamanaka factors that drive cells into a state of embryonic stem cellness is not what we want to be doing. So, and some of the genes could be causing cancer if overexpressed or not turned off. So how do you regulate their levels and control them? So we're trying to look at other approaches, including small molecule approaches that may have the ability to partially rejuvenate cells. And I think that really is the holy grail. I think in some ways, when we're focused on all these different hallmarks, many of the approaches are actually partially rejuvenating an aged cell. We just never really look at partial rejuvenation. So I think we're going to come up with different drugs and different cocktails of drugs that will show signs of non-reducing senescence, targeting other hallmarks of aging, but actually using the transcriptome, the lipidome, the metabolome, all the different omics, make a cell look younger. And then the question will be is are there safety risks doing that?
Gotcha. Yeah. it's unfortunately it takes a long time to do these studies. And
I mean, that's the problem with aging is that, animal models, if you don't work on worms and flies, take a long time. So how I got into the aging space was start to collaborate with somebody that has mouse models of accelerated aging that mimic human progerias. And so these mice age 6 to 7 times faster. So we're able to do drug treatment studies faster or assess a genetic change in these mouse models, what effect it has on aging. And then take those, what looks positive, we can take into natural aging and then hopefully eventually into people. And it may be that picking individuals that have, progerias, whether it's Hutchinson-Gilford or xeroderma pigmentosum or Cockayne syndrome, that they might be actually an orphan disease that could be these drugs, I can't say anti-aging, but drugs that extend health span might be beneficial in these individuals that are aging faster.
Mhm. Gotcha.
So that would accelerate some of these studies. So we're trying to find ways that we can identify therapeutics faster and then identify the right patient population for testing them.
Yeah. Well, I'm definitely gonna keep an eye out on your future studies and work. Before I ask my last question, where can people learn more about you and your work?
Yes. So if you look for the Masonic Institute in the Biology of Aging and Metabolism, it's called MIBAM. If you search for MIBAM at University of Minnesota, our institute's website, there's a lot of information about what's going on. We are starting, hopefully, a quarterly newsletter that you'll be able to download from our website. We've only talked about it for 6 years. We're finally launching that this fall. So we will have a newsletter that highlights accomplishments, both preclinical and clinical, highlights papers, highlights awards, et cetera, new grants. So I think between the newsletter and the website, I also have my own website at robbins.umn.edu, where, again, we have updates on papers and projects. I think it's a goal for everybody in aging research to make the public aware of what's happening, both the positive and the negative, based on high-quality science. I think science, we won't talk politics, is under attack in the U.S. at this point. So it's up for us doing aging research to get the message out about where the field is and what we've shown and what we haven't shown.
Hmm.
We're not out hawking supplements just for the sake of supplements, but trying to do well-controlled clinical studies to evaluate whether supplements work, either alone or in combination. So it's something we're all trying to do, is how to determine how to get the word out about high-level science in the aging space. But from my lab, our institute, but also around the world, how do we get this message out?
Yeah.
So we're working on new ways to convey it, whether it's podcasts, newsletters, websites, etc.
Yeah, well, I'm happy to help with that. And my last question is, what's this one piece of advice or a habit that you wish you adopted sooner?
Oh, that's a great question. Again, getting back to nutrition and exercise, it took me turning 50 before I started to think about this more. I always exercised and played sports, but then as your body gets older and your knees starts to hurt a little bit, you're not as active on the squash court. So I think I finally learned to find exercise routines in diets that I actually can follow, not just imagine, but actually follow, that are appropriate for older people. And I'm not a big pill taker, but I do take Fisetin. I have taken metformin in the past, but for whatever reason didn't continue it. But I think I'll probably be taking a few more supplements as we identify in well-controlled blinded clinical studies that they show a benefit. But I think coming up with a diet that is manageable and an exercise routine that's appropriate, that you can actually follow, is a big step forward. You don't have to go out and run marathons or bike 100 miles, but there are things you can do that can keep you fitter for longer. And I think that all has benefits as we're developing, hopefully, the first generation of, I hate the word anti-aging, but compounds that will extend health span, slow aging.
Yeah. Yeah. Well, looking forward to it too. And we have to do what is possible in that time
Yes.
and moment. So right now, this is the most effective way to slow down aging.
Well, you're such a young guy. You have time before you have to start worrying about this. So I think
Right.
by the time you get older and have to worry about the aging process, there's going to be a number of pharmaceuticals and biologics that have shown efficacy in well-controlled clinical studies. So
Well,
I
yeah.
think there's going to be a lot of hope. For those of us
Well,
who are older, we keep talking about how we're running out of time. We have to
right.
do this quickly. But the field is definitely moving forward because the quality of the science has gotten
Mm,
better. And there are true scientific studies being done and not just snake oil being sold. So the field is really moving forward and a very exciting time to be in aging research.
yeah, absolutely. Well, thanks for coming to the podcast, and it was very valuable and informative.
Well, thank you for having me. I
Yep.
enjoyed it. Thank
Yep.
you.
I'll see you around.
All right. Take care.